In recent years, hospitals have become an increasingly dangerous place for bacterial strains immune to the effects of antibiotics. The hyper-sterilized environment has given an evolutionary advantage to strains that are capable of surviving and replicating in the presence of the medicine designed to fight them. Clostridium difficile is one of these strains, catching the attention of scientists in the medical community. But what about C. difficile allows it to survive in these conditions and be so lethal? Cells use gene regulation to control which genes on the genetic material are read to become proteins. The virulent strains of Clostridium difficile had two important genetic patterns that resulted in their prevalence and severity. Virulent strains were found to encode an enzyme called Tra, which allowed for the metabolism of the sugar Trehalose. Virulent strains also had a mutation on the GRA gene that led to antibiotic resistance when symptoms were being treated in hospitals. Both of these genetic factors provided virulent strains with an advantage that allowed them to thrive and continue to be encoded to make proteins.
The development of the two most virulent strains of C. difficile, ribotypes 27 and 78, occurred via a mutation in the GYRA gene, likely during DNA replication, which led to the pathogen's ability to be resistant to fluoroquinolone antibiotics. While these strains have been around for several decades, there are a number of possibilities that could account for the increased virulence observed in hospitals recently. The strains developed from a common ancestor, but their lineages branched apart a long time ago, meaning the virulence in both strains is most likely from unrelated occurrences. To determine what makes the virulent strains so much more prevalent and severe, Collins tested the growth of different strains with different types of carbohydrates. To measure growth, a phenotype microarray was used. Phenotype microarrays contain compounds that change color when metabolized, allowing a microplate reader to monitor the metabolism by measuring the intensity of the color via light absorbance. This process revealed that the disaccharide trehalose correlated with more growth of the virulent strains than non-virulent strains. Furthermore, the virulent strains could grow in low levels of trehalose, whereas the non-virulent strains could not. Collins' experiments show that the severity of the ribotype 27 strain, causing severe diarrhea and sometimes death, can be attributed to its modified TreR protein, which allows the bacteria to perform trehalose metabolism at low concentrations of trehalose. This modified protein is a result of a mutation that occurred near the Tra binding site. To test this, ribotype 27, the virulent strain, and ribotype 53, a non-virulent strain, were placed in increasingly concentrated solutions of trehalose. Both strains contain the TreR protein and the Tra gene that is responsible for the trehalose regulation. The results showed that ribotype 27 was able to utilize trehalose at a rate 500 times higher than ribotype 53.
The overall findings of this report were that dietary trehalose enhances the virulence of strains of the bacterium Clostridium difficile. More specifically, the authors of the report suggest that the implementation of trehalose as a food additive into the human diet helped select for their emergence and contributed to hypervirulence. This conclusion is based on a few key lines of evidence. Primarily, the two strains exhibited mechanisms to metabolize trehalose prior to any major outbreaks of the bacterium. Second, the two strains have evolved distinct yet functionally similar mechanisms to metabolize low levels of trehalose, conferring upon them a selective advantage and a stronger fitness despite low levels of trehalose. Third, the experiment utilized a mouse model of C. difficile, in which a mouse was subjected to the strain, and the concentrations of trehalose were manipulated. The positive correlation between trehalose concentration and mortality observed in the mice is consistent with increased virulence of the RT027 and RT078 strains observed in human patients. The results of these experiments are described in Figure 3 of the report, which plots survival probability of test mice based on the strain of C. difficile injected. Mice infected with RT027 have a significantly higher risk of mortality when trehalose is supplemented in the diet than those with no trehalose supplementation, indicating that trehalose enhances virulence. These results ultimately can transfer over to cases of human infection. On the basis of these observations, the authors of this report propose that the widespread adoption and use of disaccharide trehalose in the human diet has played a significant role in the emergence of these epidemic and hypervirulent strains. The main author of this report, J. Collins, said regarding his study, quote, "An important contribution of this study is the realization that what we once considered a perfectly safe sugar for human consumption can have unexpected consequences," further adding that, quote, "Our study suggests that the effect of trehalose in the diet of patients in hospitals with RT027 and RT078 outbreaks should be further investigated." These reports reflect the clear importance of these findings as well as the need to further investigate how food additives, such as trehalose, shape these kinds of biological interactions.